Hello, am I clear to you? Am I audible? Shall we start?
Yes, ma'am. You can
Yes.
start.
Okay. Good morning, everyone. In our last class, we have studied about biological agents, that is, molecules with different chemistry, and where we have studied in detail about different types of the molecules, which includes neonicotinoids, toxins, spinosad, ivermectin, and then we have studied apamectin, ivermectin, and then nebulizer, petrolane, formamidins, indexotar, etc. We have studied these things in our last class, where we have studied about the importance of each molecule and their properties and its mode of action along with metabolism. Okay, and today we are going to talk about the next topic, that is about synthetic insect control agents. Okay, and here under the synthetic control agents, we are going to talk about the different types of synthetic insecticides, where we are going to study about its importance in controlling the pests, okay, with different examples. Okay, and now coming to the synthetic insect control agents. While we are studying about the synthetic insect control agents, in order to reduce the population of the insects by affecting their behavior or the genetic material or the growth. We all know, since most of the chemicals are more specific nature, they can find an important place in different programs. Though no insect chemosterilant has been either tested, used, or registered in India for the control of insect pests, these have been included to study in this particular synthetic insect control agents. Okay. And here, if you could see the name itself, it is indicating these are the insecticides that are used to control the pests. These are the control agents, which are also known as synthetic insecticides. These are chemical substances which are used to kill or to manage the population of the insect that cause threat to crops, livestock, or the human health. And these agents are critical in agricultural practices, public health programs, and urban pest control. Here you can see some of the examples of synthetic insecticides which we have covered already in our previous classes. Examples are organochlorines, organophosphates, carbamates, pyrethroids, neonicotinoids, and some of the insect growth regulators. We have covered these things in our previous classes. Okay, and today we are going to talk about the other important synthetic insecticides. These play an important role in controlling the insects. The first one under this is chemosterilants. What are chemosterilants? The name itself is indicating these are the chemicals which are used to control pest populations by inducing sterility in insects. Unlike conventional insecticides which are used to kill insects directly, chemosterilants target the reproductive system of insects, preventing them from producing offspring. And this approach can effectively reduce insect populations over time without the immediate environmental impact of killing non-target species. Hope you understand about the chemosterilants. Okay? This is the first one under the synthetic insect control pests. If you could see the mechanism of action, these work by interfering with the reproductive process of insects. and they can affect both male and females, although they are often used to sterilize males to prevent them from successfully mating with females. That's how these chemosterilians can be used to control the population of the insects. And the sterilization can occur by different mechanisms. The first one is interference with DNA, and the second one is disruption of hormonal regulation, and the third one is oogenesis. What is interference with DNA? Here, these chemosterilants interfere with DNA to cause changes in the DNA or the genetic material, which are specific to reproductive cells, and those are germ cells. Coming to the disruption of hormonal regulation, these chemosterilants affect hormones that regulate the reproduction process. So that when there is an imbalance in the hormones, obviously the reproduction process gets affected, which does not allow the organism to produce the offspring. And coming to the inhibition of spermatogenesis or oogenesis, these chemosterilants prevent the formation of either the egg or the sperm in both male and females. That's how it could able to control the population of the insects. These are the three mechanisms that are involved that occurs by the action of the chemosterilants. And coming to the next part about the chemosterilants, we are going to talk about alkylating agents and non-alkylating agents. What are alkylating agents? That means chemosterilants can act as either alkylating agents or non-alkylating agents. Those are called as alkylating chemosterilants and non-alkylating chemosterilants. What are alkylating chemosterilants? These are a class of chemosterilants that induce sterility by adding alkyl groups to DNA, proteins, and other biomolecules which are a part of the reproductive cells. And this alkaline process can lead to mutations, cross-linking of DNA strands, and which eventually leads to the inability of the cells to function or replicate properly. There are some examples which are given here. As alkylating chemosterilants, examples are methyl methane sulfonate, ethyl methane sulfonate, aziridines, and dimethyl sulfate. These are some of the examples of alkylating agents which are acting as chemosterilants. This is an extra information which I have given here. But according to your syllabus, we have to study about alphalate, tefa, and metafa. These are the alkylating agents which are acting as a chemosterilants. We are going to concentrate apart from ethyl methane sulfonate, and aziridines and dimethyl sulfate. We are going to concentrate on these things as it is given in your syllabus. And as you all know that chemosterilants are the chemicals which induces the sterility in both male and female population of the insects. Now coming to the alkylating agents, these are a particular class of chemosterilants, like non-alkylating agents. These are also used to control the insect populations by inducing sterility. Here you can see some of the examples. Alphalate, its chemical name is aziridine phosphine oxide. Please, phosphine oxide in simple form. If you could see the molecular formula, it is made up of carbon, hydrogen, nitrogen, phosphate, and oxygen, and its molecular weight is 231.20 grams per mole. And if you could see its appearance, morphology, it is a white crystalline solid, and it is soluble in water and organic solids. Coming to its action, the first one is DNA alkylation. What is DNA alkylation? The first one, what we are discussing about the under alkylating agents are alphalate. Alphalate alkylates the DNA in insect reproductive cells, which leads to changes in the DNA or the genetic material. Those are called as mutations. Not only that, Cross-linking of DNA strands also occurs by this affiliate. This leads to the destruction of the normal functioning of the cells ultimately, which causes the sterility in the targeted organisms, either the insects or the pests. What is target insects? Affiliate is effective against a range of insects, including mosquitoes, fruit flies, and other agricultural pests. Ultimately, the function of chemostimulants is to induce sterility in the pest to control the population. Okay, so how it acts? It causes mutations and it causes cross-linking of the DNA strands that ultimately leads to the normal. Ultimately leads to disrupting the normal function of the cells, especially in the reproductive cells, which causes sterility in that particular targeted organism. Did you all understand? Am I clear to you all?
Yes, madam.
Okay. Now coming to the application of this affiliate, it can be applied via direct contact or by ingestion or by environmental exposure. It is often used in controlled environments like laboratories or greenhouses. And it is used in sterile insect techniques programs where sterilized males are released into wild to mate with females, leading to reduced pest populations over time. Ultimately, our target is to control the pests, to protect the crop, to protect the human beings, to protect the environment. So, instead of killing the organisms directly, we are just controlling its population by inducing sterility. And the second one under alternating chemostimulants is HEPA, that is this phosphine oxide. If you could see its chemical structure, chemical formulae, chemical structure of chemical formulae, it contains carbon, it is made up of carbon, hydrogen, nitrogen, oxygen, and phosphate. And its molecular weight is 173.15 grams per mole. It is stateless liquid and it is miscible with water and organic solvents. Coming to the action of HEPA, these forms cross linkages within DNA molecules and disrupting the functioning processes in the cells, which causes sterility. Okay, in what way it is causing disruption? It is not allowing the DNA to replicate. It is not allowing the DNA to form the protein by central dogma of molecular biology. When it is not forming the protein, the required metabolism does not occur in that particular cell, but eventually the cell function will be altered and ultimately that leads to sterility in the organism. And it is effective against mosquitoes and houseflies. And this HEPA can be administered through direct contact or environmental exposure, and it is also used in laboratory settings for research on insect sterility. Similar to affiliate, HEPA is used in SIP programs to control pest populations by releasing sterilized males into the environment. Whatever the things which you are seeing here with respect to HEPA is similar to the things which you have seen in affiliate. Okay. I'm coming to the third one under alternating chemostimulants. If you could see here, the third one is METHEPA, whose chemical name is this phosphine oxide. There is just a slight variation is there is seen in METHEPA when compared with affiliate and HEPA. Okay, here also it is made up of carbon, hydrogen, nitrogen, oxygen, and phosphate, whose molecular weight is 215.23 grams per mole, and it is colorless to pale and non-liquid. It is soluble in water and organic solvents. These METHEPA alkalides the DNA in insect reproductive cells and causing mutations and also inhibits the cell division. This leads to sterility in trigger insects. Once we complete these things, we'll talk about what are mutations and what is the central molecular biology to understand the things very easily. Let's come into the target insects. These metapod targets, mosquitoes, fruit flies, and other agricultural pests. And this can be applied through direct contact, ingestion, or environmental exposure. And it is commonly used in controlled environments for sterility research and used in set programs where sterilized males are released to reduce the population of pest insects over time. This is all about the alkylating thermosterilants. Here in the alkylating thermosterilants, we have covered three important things. The first one is acylate, second one is stepa, and the third one is metapod. These are the different types of alkylating thermosterilants, which are acting as a valuable tools in the management of pest. In the insect pest management. Here, by inducing sterility in target insect populations, and these alkylating agents offer an environmentally friendly alternative to conventional insecticides, and their use in sterile insecticide programs demonstrate their potential to sustainably control pest controls, although careful application and monitoring are essential to maximize their effectiveness and minimize their resistant development. Okay, this is the importance of acylate, tepa, and metapa. How exactly these are targeting the reproductive cells? These are targeting the DNA, that is the genetic material. The genetic material plays a major important role in controlling the organism. The personality and metabolic activities of that particular organism. Everything depends upon the genetic material, which is called as a DNA, deoxyribose nucleic acid. Here, if there is any alteration in the DNA, as we have studied in our lower classes, sometimes changes may occur by the synthetic insecticides like aflatoxin or metapod. But sometimes environmentally also spontaneously you could see some changes in the DNA. But here we are inducing the changes in the DNA in order to control the pests. These are not spontaneous. These are induced mutations. There are two types of mutations. The first one is spontaneous mutation and the second one is induced mutations. Spontaneous mutations occurs in nature, and the induced mutations occurs either by physical methods or chemical methods or biological methods. But here we are using the chemicals to induce mutations in the DNA of the insects. Especially we are targeting the reproductive cells, not to provide the offspring. That means we are controlling the population. So this is all about the alkylating thermosterilants. Hope it is clear, right? Did you all understand?
Yes, ma'am.
Is there any doubt in the alkylating thermosterilants? Hello? Hello? Is there any doubt?
Yes. No doubt, ma'am.
Everything clear? Shall I start with the next topic?
Yes, ma'am.
Okay. Now we are going to talk about the non-alkylating thermosterilants. Can you see here about the non-alkylating thermosterilants?
Yes, madam.
Okay. Now we are coming to the non-alkylating chemostimulants. These are the class, a particular class, as you have seen previously, like the alkylating chemostimulants. And these acts as a chemical molecules which induce sterility in insects without the use of alkylation. Okay, and these agents utilize various mechanisms to interfere with the reproductive processes of insects, making them effective tools in integrated pest management. So, under this, we are going to talk about phosphoramides. This is the first one that we are going to study under non-alkylating chemostimulants. These phosphoramides are a class of non-alkylating chemostimulants which induce sterility in insects by interfering with their reproductive process. There are two examples which we are going to study under phosphoramides. The first one is hempa, and the second one here is thiohempa. Here it is given. This is, yes, thiohempa. Okay, these are the two things which we are going to study under phosphoramides. First one is hempa. If you could see the hempa, its chemical name is hexamethyl phosphoramide, whose structure is made up of carbon, hydrogen, nitrogen, oxygen, and phosphate. And its molecular weight is 179.20 grams per mole, which is a colorless liquid, and it is soluble in water and organic solvents. This hempa, which is a non-alkylating chemostimulant, interferes with DNA replication and transcription process in reproductive cells, which leads to mutation and reproductive failure. It can also inhibit enzymes which involves in reproductive process, and leads to the sterility. Coming to the usage of hempa, it is effective against different types of insect pests like mosquitoes and agricultural pests. And this hempa can be applied to direct contact or environmental exposure, often used in controlled environments such as laboratories or greenhouses. What are the advantages of hempa? It is proven to be effective in reducing pest populations by inducing sterility. Generally, it has a lower environmental impact compared to traditional insecticides. Coming to the disadvantages of using hempa, it can be toxic to non-target organisms. Maximum all the insecticides are toxic to non-target organisms and humans if not handled properly. So we have to take proper measures while we are using different types of the insecticides to control the pests. Okay. And the second one under phosphoramides is thiohempa. This is maximum similar to the type of the hempa, whose chemical structure contains carbon, hydrogen, nitrogen, sulfur, and phosphate, and its molecular weight is 271.35 grams per mole. And it is a low crystalline solid, soluble in water and organic solvents. If you could see the function of thiohempa, it causes cross-linking in the genetic material of the target organism, ultimately leads to the changes in the cellular processes to cause sterility. It can also interfere with various metabolic pathways which are essential for reproduction. Coming to the usage, it is effective against various insect pests, mosquitoes, flies, and agricultural pests. And it can be applied via direct contact, ingestion, or environmental exposure, often used in experimental setup to evaluate its effectiveness in inducing sterility. And what are the advantages of using thiohempa? It is effective in reducing populations of a wide range of insect pests. Generally, it has a lower persistence in the environment compared to some traditional insecticides. And if you could see the disadvantages, insects may develop resistance over time, reducing the long-term effectiveness of the compound. Like other chemostimulants, these can be concerns about toxicity to non-target organisms and humans. Always, that is the reason always we should not use the same type of the insecticide to control the pests. Often we have to change the type of the insecticide to control the pests so that it does not develop any resistance towards that particular insecticide. And coming to the application, we have to spray or apply this particular phosphoramides directly to areas where the target insects are present. And we have to incorporate this compound into beds that are consumed by the target insects. Releasing the compound into the environment where insects are known to breed and feed. If you could do these things, we could able to sterilize the insects to control its population. And coming to the integration into pest management program, using sterilized males in combination with these compounds reduces the pest population. Combining chemostimulants with other pest control methods to enhance overall effectiveness and sustainability. This is all about phosphoramides. In phosphoramides, we have studied about two things, hempa and hyohempa. These are the examples of phosphoramides. These are significant phosphoramides which we have studied, and these are used as chemosterilants that are used to control the insect population by inducing sterility. And they offer a targeted approach in order to manage the pests or to control the pests with reduced environmental impact compared to traditional insecticides. And the other thing what we have studied under this is these compounds can be a valuable components of integrated pest management strategies which control the pests and contributes to sustainable and effective pest control. However, their application requires careful handling and monitoring to ensure effectiveness and minimize risk to non-target organisms along with the humans. This is all about phosphoramides. Phosphoramides, under phosphoramides, we have studied about hempa and hyohempa. Hope it is clear for you. Is it clear?
Yes.
Did you understand?
Yes, madam.
Okay. Under non-alkylating chemostimulants, we have covered phosphoramides. And coming to the second one, under non-alkylating chemostimulants, melamides. These are also a class of non-alkylating chemostimulants that contains a heterocyclic organic base, which is called as a melamine. That is the reason it is named as melamides. It itself is a primer of cyanamide with 135 triazine skeleton. It is a notable for its high nitrogen content, which makes it valuable in various industrial applications. If you could see the chemical formulae, it has carbon, hydrogen, and nitrogen. It contains 3 cyanamide groups, which are connected with aromatic triazine ring. If you could see the structure, it is melamine unit properties such as it shows thermal stability and resistant to wide range of chemicals. If you could see the melamine, it is white crystalline solid at room temperature. It is odorless. And it has a melting point of about 354 degrees Celsius. It is slightly soluble in water. Coming to the chemical properties, it is known for its ability to form strong hydrogen bonds. It can form stable compounds with formaldehyde, resulting in resins used in various applications. And this melamine is also known for its high nitrogen content, which contributes to its fire retardant properties. Coming to the examples of melamines, the first one is HMM, that is hexamethyl-melamine. This is also a non-alkylating chemostimulant. Hello? Hello? Any doubt? Is there any doubt?
No, ma'am. No, ma'am.
Okay. If you find any difficulty, please let me know. Okay. Coming to the examples of melamines, The first one is HMM, which stands hexamethylmelamine. It interferes with DNA synthesis and repair mechanisms, which leads to sterility. That means DNA synthesis means replication. During replication, there are some enzymes which involve while forming the bonded strand, using the parental strand as a template, there will be a chance of incorporating a wrong nucleotide in the daughter strand. At that time, there are some enzymes like DNA polymerase 1 should come and repair, that means remove the wrong nucleotide and incorporate the correct nucleotide. Okay, so these things will not occur with interference of hexamethylmelamine. This you are going to see in the insects. That means this hexamethylmelamine chemical, when insect get exposed to this chemical, it interferes with DNA synthesis and repair mechanisms. When there is no proper DNA, when there is no proper DNA strands that can be produced for the survival of the organism, that leads to the sterility. It is, and next, coming to its usage, it is used in research settings to study sterility effects in insect populations. And the second one under melamine is triaminotriazine. This is the example of melamine, which inhibits the reproductive hormone synthesis and function, and it is applied in experimental setups to manage pest populations. This is all about the melamine. And the third one under non-alkylating chemosterilants are dithiotarbamates and dithiazonium salts. These are the things which we are going to study. And before we talk about this, these melamines are active against houseflies especially. Under melamines, we have studied about what is its structure, what are the properties, and what are the examples of the melamines. These are lots of alkylating agents. These are active against houseflies. Coming to the third, dithiocarbamates and dithiazonium salts. Coming to these things, if you could see, these are active chemosterilants, which are used in controlling the pests. And the examples of these are xylem, thyrum, aminomethyl, dithiazonium salts. These are the three types which are going to see under dithiobutans and dithiazonium salts. Coming to the xylem, which disturbs the enzyme function critical for the reproductive process, and it is used in pest control. Coming to the thyrum, it inhibits enzyme systems leading to the reproductive failure, applied in agriculture to control seed and soil-borne pests. And coming to the third one under dithiobutans and dithiazonium salts, third example is aminomethyl dithiazonium salts. It disturbs metabolic pathways which are essential for reproduction, and they are used experimentally to evaluate its effectiveness in reducing sterility. This is all about dithiobutans and dithiazonium salts. Okay, and coming to the miscellaneous compounds that we are going to study under alkylating chemosterilants. Given here, listed here, the first one is aminopterin, and the second one we are going to talk about 5-fluorophilic acid, and the third one we are going to talk about nitrogen mustard, and the fourth one we are going to talk about anthomycin, and the fifth one asparaginase. These are the things which we are going to study under miscellaneous compounds. Coming to the first one, here in this group of non-alkylating reagents, we are going to talk about aminopterin. Coming to the chemical structure of aminopterine, it is made up of carbon, hydrogen, oxygen, nitrogen, and its molecular weight is 440.41 grams per mole, and it is a yellow crystalline powder soluble in water and organic solvents. Coming to its mode of action, it inhibits dihydrofolate reductase, which is an enzyme required for the synthesis of nucleotides. When there is no synthesis of nucleotides, there will be no replication of the DNA. When there is no replication of the DNA, obviously there will be no cell division. That ultimately leads to sterility. Okay. Coming to its usage, it is used in research to study sterility effects on various insect pests. And it is typically administered through ingestion or environmental exposure. Coming to the advantages of aminopterine, these are specifically used to disrupt reproductive process in the insects and valuable in environmental experimental studies on insect reproduction. And it can be toxic to non-target organisms and requires careful handling. This is about aminopterine. We are going to talk about the second part, 5-fluorouracil. What is 5-fluorouracil or 5-fluorotate acid? If you could see its structure, it is made up of carbon, hydrogen, fluorine, nitrogen, and oxygen. And its molecular weight is 130.08 grams per mole, and it's a white crystalline powder soluble in water and organic solids. Coming to its mode of action, this 5-fluorouracil inhibits time-delayed synthase, which is an enzyme. When this gets inhibited, which disrupts the DNA synthesis and repair mechanisms, leading to cell death and sterility. Coming to its usage, it is applied in research settings to assess its potential in inducing sterility in pests, and it is administered via direct contact or ingestion. It is proven to disrupt reproductive processes effectively, and it can be used in various experimental settings. And coming to its disadvantage, we have to use this very carefully to avoid harm to non-target species. This is all about 5-fluorouracil. And the third one is nitrogen mustard. What is nitrogen mustard? Which is a mitotic poison. Okay. Coming to its chemical structure, it is made up of same carbon, hydrogen, chlorine, and nitrogen. And its molecular weight is 159.03 grams per mole. It is a colorless to pale yellow oily liquid, soluble in water and organic solvents. If you put its action of the nitrogen mustard, okay, it targets when it forms the toxin in the DNA strands that prevents replication and transcription that leads to the cell death and sterility. Okay, and it will be applied through direct contact or environmental exposure. Coming with the advantages of using this nitrogen mustard, it is effective in inducing sterility by disrupting DNA function, and it is valuable for studying the effects of DNA alteration on reproduction. Coming to the disadvantages, we have to handle it properly to avoid harm to non-target organisms. And the next one, fourth one, under miscellaneous compounds is anthromycin. This anthromycin is an antibiotic. This sterilizes few of the flies like Drosophila and housefly. Okay, and if we could see the chemical structure, it is made up of carbon, hydrogen, nitrogen, and oxygen, and its molecular weight is 289.33 grams per mole. And it is a yellow crystalline powder soluble in water and organic solvents. Coming to the action, this anthromycin intercalates with the DNA to disturb the replication and transcription process, which leads to the death of the cell and sterility. And this anthromycin is administered via direct contact or ingestion, applied in research to assess its effectiveness in inducing sterility. Coming to the advantages of using anthromycin antibiotic, which is proven to disturb reproductive processes effectively, valuable for studying the effects of DNA intercalation on insect reproduction. And this anthromycin can be positive non-target organisms if you are not going to use properly. And the last one, fifth one is aspergillus. This aspergillus is a chemosterilant, which is an enzyme. If you could see its structure, it doesn't have a fixed chemical structure. It is an enzyme. All enzymes are protein, as we already know. And its molecular weight depends upon the source from which we are isolating it. And if you could see its physical state, it is white to off-white powder, and it is soluble in water. Coming to the mode of action, this aspergillase enzyme depletes the amino acid aspergine. You all know that protein is made up of amino acids. Amino acids are the building blocks of the proteins. And this aspergillase enzyme depletes the amino acid aspergine, which is essential for protein synthesis and cell survival. If there is no protein synthesis, the required metabolic pathways will be altered, and which leads to the reproductive failure and the death of the cell. Coming to its usage, we have this should be administered through ingestion or injection. And the advantages of aspergillase is it targets a critical metabolic pathway in insects to stop the formation of the proteins, and it is valuable in experimental studies on insect metabolism and reproduction. And coming to its disadvantages, we have to use it very carefully. Coming to the advantages and disadvantages of non-alkylating agents. These are often less toxic and persistence in the environment compared to traditional insecticides. And they can specifically affect the reproductive process without killing non-target species, suitable for integrated pest management, reducing the need for conventional insecticides. Coming to the disadvantages of using non-alkylating chemosterilants, there are potential for insects to develop resistance, though this varies by compound and species. They require precise application and monitoring, which can be more complex than using conventional insecticides. Initial research and implementation can be expensive due to the need for specialized compounds and growth. These are around non-alkylating chemosterilants. In non-alkylating chemosterilants, we have studied about few things. The first one is phosphoramides, second one is melamines, third one is dithiobutirates and dithiazolium salts, and the fourth one is miscellaneous compounds. And phosphoramides, we have studied about compound and biocompound, and melamines, we have studied about importance and dithiobutirates and dithiazolium salts. Also, we have studied only two things. And coming to the miscellaneous compounds, we have studied about 5 important things. Okay, all these are involved in altering the DNA synthesis or replication or transcription process or translation process directly or indirectly to cause the inhibition of different metabolic pathways in order to induce sterility in the target organism. Okay. Hope it is clear to you all, right? Hello?
Yes, madam.
Amma, is there any doubt that which we have completed?
Madam, is this dithiobutirates same like dithiocarbamates?
Yes. Yes. All are pesticides. More or less slight changes you would see in these compounds. Whatever we have studied previously, maximum they all Their function is same, slightly they are different. That's it.
Okay, madam.
Okay.
Okay, madam. Thank you.
So by this we have completed the insect sterilized. Okay. The name itself is imitative, which causes sterility to the insects. These are the chemicals which causes sterility to the insects. So these are called as insect chemosteroids. Under this, we have studied about alkylating agents and non-alkylating agents. Okay. And turning to the alkylating agents, um, we have covered acrylate, Kappa, and metapa. And coming to the non-alkylating chemosteroids, we have studied about phosphoramides, melamines, dithiobutirates, and dithiosodium salts, and about pterin, asparginase, all these things we have covered. Okay, and now we are going to talk about insect growth regulators. Okay, here we are going to study about few of the chemicals, which how these chemicals regulate the growth of the insects. Okay, if we could able to regulate the growth of the insects, somehow we could able to manage the pests. Okay, so under the insect growth regulators, we are going to cover a few important things. If you could see the definition of insect growth regulators, these are the chemicals which controls the insect population by interfering by inhibiting its growth and development. Okay, in the insect growth regulators, there is a specific class which is called as tyrosine synthesis inhibitor. These tyrosine synthesis inhibitors are a specific class that targets the synthesis of tyrosine. We all know that it is a exoskeleton which you are seeing on the outside of the insects. Okay, and by stopping this tyrosine synthesis, these inhibitors prevent insects from developing properly, leading to mortality or deformities. This tyrosine protects the insect. If there is no proper development of this exoskeleton, which is a part of the insect, that leads to the mortality. The first one is tyrosine synthesis inhibitor. What is tyrosine synthesis inhibitor? This prevents the formation of the tyrosine, which is a polysaccharide, which is a very important molecule to form an exoskeleton as well as insect exoskeleton as well as the duct lining. This tyrosine provides structural integrity, and without it, insects cannot properly mold or develop. It leads to the death of the insects. So, what are the inhibitors of tyrosine synthesis? There is an example which I have listed here. The first one is benzoyl phenylureas. These are a class of insect growth regulators that acts as tyrosine synthesis inhibitors. They interfere with the formation of tyrosine, a key structural component of the insect exoskeleton, thereby preventing proper molting and development, which results in the death of the insect. Here I have given some of the common examples of benzoyl phenylureas. Diflubenzuron, lufluron, novaluron, flufenoroxin, hexafluoride. These are the different types of benzoyl phenylureas. Coming to the structural components which you are going to see in benzoyl phenylurea. If you could see its structure, chemistry of it has a benzene ring and a phenyl ring which gets attached to the urea molecule. Coming to its solubility, it is slightly soluble in water, but more soluble in organic solvents. And these benzoyl phenylureas are stable under normal conditions, but can decompose at higher temperatures or under extreme pH conditions. And the biological activity of benzoyl phenylureas Such tithing synthesis cellulose, which are highly dependent on a specific substance attached to the benzene and phenylurenes. If there is any modifications in the benzyl phenylurea, that will enhance the specificity and potency against the target organism. This is about the activity and the chemical and the physical properties of benzyl phenylurea. Coming to its action, these benzyl phenylureas target the insect growth regulators, especially they target the synthesis of exoskeleton chitin, which is a vital component of the bacterial and insect exoskeleton. Understanding the mode of action provides insight into how we effectively control insect populations by disrupting their development and modeling process. We all know that chitin plays a major important role in protecting the organism. So, if there is any change in the chitin formation, obviously the organism leads to the death. Now, coming to the mechanism, how exactly it controls the pest by inhibiting the formation of the chitin. The first one is inhibition of chitin synthase enzyme. This is an enzyme which is responsible for polymerization of N-acetyl glucosamine units into chitin, which is a polysaccharide that forms a critical structural component of the insect exoskeleton and gut lining. Coming to the mechanism of inhibition, this benzyl phenylureas inhibit the activity of chitin synthase that prevents the enzyme from catalyzing the formation of chitin. This inhibition occurs at the biochemical level, where benzyl phenylureas bind to the enzyme or interfere with its substrate binding sites, blocking the polymerization process. And the second one, there is interruption of exoskeleton formation. Coming to the interruption of exoskeleton formation, the chitin provides mechanical strength and rigidity to the insect exoskeleton, protecting the insect and providing a framework for muscle attachment. In the gut lining, chitin forms a protective barrier which aids in digestion and prevents damage from ingested materials. Coming to the effects of chitin synthesis inhibition, without chitin, the structural integrity of the exoskeleton is compromised. The insect cannot produce a functional exoskeleton during molting, leading to deformities and structural weakness. Coming to the third one, disruption of molting process. That is, molting is a process by which insects shed their old exoskeleton and form a new. Successful molting requires the synthesis of new exoskeleton, which relies on the production of chitin. What is the impact of benzyl phenylurea? Chitin synthesis. Benzyl phenylurea prevent the formation of a new exoskeleton, disrupting the molting process. Insects are unable to shed their old exoskeleton properly and leading to molting failure. And the first one under the mechanism is developmental arrest and mortality. Insects undergo multiple developmental stages where they evolve to grow and develop into the next life stage. Coming to the consequences of chitin synthesis inhibition, benzyl phenylureas cause developmental arrest at various stages as the insect cannot progress to the next stage without successfully molting, without shedding the exoskeleton. And the inability to molt properly leads to the death of the insect. And here I have given some of the examples of benzyl phenylureas, which I have given previously, which I have listed. Diflubenzuron, which prevents chitin formation in larvae, causing the death of the organism. Glufeniron, which interferes with chitin synthesis, prevents the development in the eggs and effective control sleep populations. Novuliron, which inhibits chitin synthesis in various insect pests, leading to the death of the insect during molting stages. Fluphenoxidone disturbs the formation of the exoskeleton in caterpillars and beetles, causing developmental failures. Next, hexafluoride used primarily for termite control, that is chitin synthesis leading to the collapse of termite colonies. Coming to the advantages of benzylphenylurea, these specifically target chitin synthesis, which is unique to insects and some other arthropods, minimizing impact to non-target organisms, including humans and vertebrates. These have relatively low toxicity to mammals and beneficial insects when we use properly. By disrupting a critical physiological process, benzylphenylureas effectively reduce insect populations and prevent the establishment of resistant generations. There are some challenges and considerations. Sometimes the insect shows resistance to benzylphenylureas over time. Sorry. Coming to the challenges, insects may develop resistance to benzylphenylureas over time, necessitating the use of resistance management strategies such as rotating with other classes of insecticides. Some of the benzylphenylureas can persist in the environment for a longer period, which may affect non-target insects sometimes. So, careful application and adherence to recommended usage guidelines are essential. These are used to control the pests, to manage the insect populations. They can be applied in different forms, including sprays, dust, and baits. And their use is regulated to ensure environmental safety and minimize the development of resistance in target insect populations. This is all about benzylphenylureas. Here, in benzylphenylurea, we have studied its chemistry and its function and some of the examples of benzylphenylurea. Here, we have seen that this benzylphenylurea is effective insect growth regulator, which disrupts the synthesis of chitin, which is a very important component for the insect exoskeleton. And by inhibiting the chitin synthase enzyme, this benzylphenylureas prevent molding the insect, which ultimately leads to the death of the insect population. And if you could go to the next level, sometimes because of its continuous persistence in the environment, it may target the non-target organisms. So proper handling must be there. And it shows it is effectively used to control the pests. And though it shows low toxicity to non-target organisms, but proper management should be there, proper usage must be there. This is all about benzylphenylureas. Okay, if you could see its chemistry, it is made up of benzyl and phenyl, which is attached to the urea molecule. And this is all about the benzylphenylureas. Hope it is clear for you, right?
Yes, madam.
Is there any doubt with the benzylphenylurea?
No.
Am I clear?
Yes, madam.
Okay. And we are going to see the next part on the insect growth regulator. The first one, on the insect growth regulator, we have studied about chitin synthesis and beta. And now we are going to talk about buprofezin. Coming to the buprofezin, it is an insect growth regulator, as we already know. It is used to control sucking insects, aphids, whiteflies, etc. Here we have given the chemistry of it. It is made up, if you do proceed structure, it is made up of carbon, hydrogen, nitrogen, and oxygen. And its molecular weight is 301.44 grams per mole. And if you do see its structure, it has a distinctive chemical structure with a triazine ring and entered butyl group. In my next class, I will try to share the structures of all these compounds which we are dealing with now. And coming to the mode of action, this bupropion has by inhibiting titin synthesis because this we are studying under titin synthesis inhibitor. So it has been inhibiting titin synthesis, especially titin synthase enzyme. This disturbs the formation of the exoskeleton titin. Especially this does not allow the formation of the titin, which ultimately leads to the improper exoskeleton formation during bonding process, which leads to the death of the which leads to the embryo development and death of the organism. Coming to the uses, these bupropion titin synthesis inhibitors are effective against certain insects, and they are used in a variety of crops, including vegetables, fruits, cotton, and ornamentals. And they are applied as a spray to foliage or as a soil treatment, depending on the pest, target pest and crop. Coming to the advantage, they selectively target insects that feed on plant sap, minimizing the impact on beneficial insects and mammals. It generally shows low toxicity to humans and animals with favorable environmental safety profile when used according to label instructions. And it provides effective control of target pests by disrupting their growth and development cycles. This is all about the importance of bupropion. Coming to the applications in pest management, it is often used as a part of IPM programs to reduce reliance on broad spectrum insecticides and preserve natural pest control mechanisms. And the application time is very much crucial to target susceptible stages of insects' life cycle. This is all about bupropion. By this, we completed titin synthesis inhibitors. Under bupropion, we have studied about its chemistry and its mode of action. These are often used in agriculture to control sucking insects, which sucks the sap of the plant. This chemical, this synthetic insecticide, bupropion, targets the enzyme titin synthase to disturb the formation of titin synthesis, and its selective action and relatively low environmental impact made it suitable for integrated pest management strategies. However, maintaining pesticide propagation and resistant management are essential to maintain its effectiveness over time while minimizing adverse effects on the environment. So this is all about bupropion, which is a part of titin synthesis inhibitors. Till now, we have studied about the insect growth regulators, how the growth of the insect gets regulated by inhibiting the titin synthase enzyme by benzophenylurea and bupropion. Is that clear to you all? Hello? Am I clear to you? Shall we go to the next part?
Yes, madam, will continue.
Okay. And insect growth regulators, the next part is juvenile hormone limits. Juvenile hormone limits. Only two parts are there, which we are going to study about these things now. The first one here is juvenile hormone limits. These are also insect growth regulators, which limit the action of juvenile hormones in insects and disrupting their normal development and reproduction. There are two common juvenile hormone mimics. Examples are methoprene and phenoxyetol. The first one is methoprene, and the second one is phenoxyetol. Coming to the methoprene, it is an analog of juvenile hormone, especially juvenile hormone 3. If you could see its action, this juvenile hormone, methoprene, binds to juvenile hormone receptors and activating the signaling pathway. That means here, this methoprene is acting as a ligand in order to bind to the juvenile hormone receptor to activate the signaling pathway that controls the growth of the insect and the development. Coming to the next function, this methoprene juvenile hormone interferes with the metamorphosis process in the insects, preventing the transition of the insect from larval stage to the pupal stage. It is inhibiting the adult dimensions. That's how it would be able to stop the growth of the insects. And these are effective against mosquito larvae, fleas, and other pests which undergo metamorphosis. This is about methoprene. And the second one is phenoxyetol. If you could see the phenoxyetol, it is also a juvenile hormone mimic like methoprene. Okay, and if you could see its chemical structure, it's a carbamate insecticide, which acts as a juvenile hormone mimic. This hormone, this phenoxyetol hormone, disrupts the insect's development process, like methoprene. Not only that, it also interferes with the insect growth at the metamorphosis stage without leaving it to convert into the adult stage. Okay. And it is used against various insects like lepidopteran such as caterpillars as well as mosquitoes. If you could see the advantages of using this juvenile hormone mimics, example methoprene and phenoxyetol, they have selective toxicity towards insects while posing no risk to mammals and other non-target organisms. They are considered safer alternatives to broad-spectrum insecticides due to their targeted mode of action and reduced impact on beneficial insects. And coming to the resistant management, rotating or alternating these IGRs with other insecticides can help delay the development of resistance in target pest populations. These can be applied as sprays, baits, or in a formulation suitable for larval habitats and or on plant surfaces where pests are active. This methoprene and phenoxyetol are important components of IPM programs offering effective control while minimizing the environmental impact. Application of these juvenile hormone mimics, timing is critical to target vulnerable stages of insect development when they are most susceptible to the effects of these IGNs. This is all about the juvenile hormone mimics. And the third one is pyriproxyfen. This is also another example of the juvenile hormone mimic. Coming to the methoprene and phenoxyetol, these are the modern pest management mimics insecticides, which disrupts the insect growth and development. And their ability to mimic natural hormones and selectively target pest species make them valuable tools in integrated pest management strategies and promoting sustainable growth agricultural practices while reducing the reliance on broad-spectrum insecticides. However, we have to get proper guidelines and we have to use these juvenile hormone mimics carefully. We have to apply the techniques properly in order to maintain their efficacy and environmental safety over the time. This is about the methoprim and phenoxy tar. And the next one we are going to talk about here is pyriproxyfen. This is also an insect growth regulator, which is one among the juvenile hormone mimic, primarily used in agriculture and public health for controlling insect populations. If you could see its structure, it is made up of carbon, hydrogen, and nitrogen and oxygen, whose molecular weight is 321.37 gram per mole. Pyriproxyfen belongs to the class of pyridine-based chemicals, which is a derivative of phenoxyether. If you could see its mode of action, this pyriproxyfen mimics the action of juvenile hormones in insects, especially disrupting the growth and development. It interferes with the metamorphosis process, inhibiting the development of larvae into the pupa and adult. This disruption leads to morphological abnormalities and sterility in adult insects. This can also affect adult reproduction by disrupting egg development and reducing egg viability. Coming to the uses of this pyriproxyfen, it is effective against a wide range of insect pests like mosquitoes, flies, etc. It is used in agriculture for crop protection and in public health for controlling vector-borne diseases. They are available in various formulations, including sprays, granules, and insecticide-treated materials for specific applications. Coming to the benefits of pyriproxyfen, it exhibits low toxicity to mammals and other non-target organisms, making it a safer alternative to broad-spectrum insecticides. It has a minimal impact on beneficial insects and ecosystems when used according to label instructions. And it provides residual activity offering long-lasting control to insect populations after application. Rotation with other classes of insecticides helps to manage resistant development in target pests. Coming to the usage of pyriproxyfen in pest management, it is integrated into IPM programs to reduce reliance on conventional insecticides and to target specific life stages of insect pests. It is used in mosquito control programs to reduce populations of disease vectors and prevent outbreaks of disease like malaria and dengue. It is applied to crops and plants to control agricultural pests, primarily those vulnerable during larval and new stages. This is all about pyriproxyfen. It is a part of phenoxyether, which is playing a major important role in controlling the growth of the insect and the development. Okay, it disrupts the life cycle of the insects and it reduces the population of the insects effectively. Is that clear?
Yes, madam.
Okay, and the last one which we are going to talk here is tebufinoside. Coming to the tebufinoside, which is also an insect growth regulator, which acts as a addition agonist. Addition agonist. Here you can see the chemistry of tebufinoside. It is made up of carbon, hydrogen, nitrogen, and oxygen, whose molecular weight is 352.47 grams per mole. And this tebufinoside belongs to benzoyl class of chemical.
Excuse me, madam.
Yes, someone.
What is the meaning of addition agonist?
Addition agonist is a insect growth regulator, basically. And we know about agonist and antagonist, right?
Yes, madam.
Coming to the especially, first I'll try to explain agonist and antagonist. If you see the agonist ones, we all know that these are the chemicals or the substances which acts as a ligands and bind to the receptor in order to activate the biological response. Okay, understand?
Okay, ma'am.
And
Okay, ma'am.
antagonist, these are also the chemicals or the substances which they also binds to the receptor, but they does not activate the signaling pathways. Okay, when they are not
Okay, ma'am.
signaling pathways. Obviously, it involves in blocking or inhibiting the action.
Okay, madam. Thank you. Understood, madam.
These are the two which we have studied. The first one is agonist, and the second one is antagonist. What is an agonist? Which is a chemical that binds to the receptor and activates the signaling pathway for the biological response. Examples: hormones, gas. Coming to the antagonist, it is also a chemical. It also goes and binds to the receptor, but it does not activate the signaling pathway. That means it will inhibit it. Okay.
Okay, madam. Thank you.
Now coming to the adjacent agonist, like what we discussed previously, it is also a compound, okay, which mimics the adjacent. What is this adjacent? Which is a hormone. But all the things which we are studying here is a hormones. Just now we have studied about the juvenile hormones. Okay, like that, this adjacent is also hormone which you see in different insects. Okay, which helps in the formation of, which helps in the conversion of the larval stage into the adult stage, that is metamorphosis or molding, etc. Okay, and this one, as it is a hormone, as it is acting as a ligand, okay, as an agonist, it goes and binds to the a specific receptor made for adjacent hormone. When it goes and binds to the receptor, it activates the signaling pathway in order to allow the cell to go for biological processes. Okay? Did you understand? Is it clear or is there any doubt? Am I clear? Hello?
Please pardon it, ma'am.
Shall I continue or shall I repeat the same?
Please repeat it once again, ma'am.
First one, agonist. What is an agonist? It's a chemical substance which acts as a ligand, goes and binds to the receptor to activate the biological response in the cell. Okay?
Okay, madam.
What is an antagonist? Which is also a chemical that will go and bind to the receptor, but if it is an antagonist, it will block the functioning of, signaling of that particular receptor. Okay? It dampens action. Okay? That is the antagonist. Now, what is an adjacent agonist? What is agonist? You already know it is a chemical that will go and bind to the specific receptor to activate the signaling
Signaling pathway.
for the biological process. Now, here, adjacent is a hormone, okay, which you see in different insects. Okay, this hormone plays a major important role in the formation of the protein. Formation of the exoskeleton. Not only that, it also involves in the metamorphosis function. Okay, if there is any problem with this hormone, in the insect there will be no molding, proper molding, there will be no proper metamorphosis. When this adhesion goes and binds to the receptor, adhesion is an agonist. Okay, it's a hormone. When this goes and binds to the receptor, it activates the cell, it activates the signaling pathway, it activates the cell for the rest of the things. Suppose take an example. Here, in all the previous slides, we have studied about the DNA transcription process. We have studied about DNA synthesis, that is replication, all these things. So, this is the receptor which involves in activating the cellular processes for the development. Okay?
Okay, madam.
Understand?
Yes, madam. Understood.
Okay. Now we are going to talk about the table phenolzide, which is acting as a agonist, which is an example of adhesion agonist. Okay. This mimics the action of adhesion, which is a hormone that regulates molting and metamorphosis in insects. By binding to adhesion receptor, this tebuphenoxide triggers premature and uncontrolled molting, leading to the death of the immature stages. This is the mechanism what I tried to explain just now, and which ultimately leads to the destruction of the insect development. Not only that, it shows its effect on insect growth by interfering with the synthesis and release of adhesion, thereby preventing the formation of the insect exoskeleton and leading to deformities and mortality. This is all about tebuphenoxide, which is an example for adhesion agonist. Uh, it disrupts the formation of title, and it also inhibits the growth of the insect. It provides effective control of the insects, and it shows less effect on the environment. Okay, this is all about your part for today. By this, we have completed insect growth regulators. Okay, is it clear to you? Is there any doubt here?
Yeah, ma'am. No doubt.
This is all about insect thermosterilants and the insect growth regulators. And in our next class, we are going to talk about the rest of the things which is there on synthetic and insect control agents. Okay. And another thing I want to tell you today, we call your son regarding sharing the notes. Today, I will most probably I'll be to your classroom where I try to share the notes for the topics which we discussed in our previous classes. Okay.
Okay, madam. Thank you.
Okay.
ma'am. Thank you.
Shall we close for the day?
Yes, ma'am.
Okay, ma'am. Thank you so much. Take care.
Thank you.